Hi everyone, I'm Andy. Today I would like to talk about reversing the life cycle, Medusa transforming into polyp and cell transdifferentiation in Turritopsis nutricula. So what is a Medusa? It's also called jellyfish, and they usually live short lives. Most of them live like days to few months, and they have the life cycle which includes sexual and asexual phase. This is this is the sexual phase. They share the same DNA. Okay. And you can see from an egg, it becomes a planula. Planula is like a ball with hair. With this hair, you can go somewhere to land. Then it becomes polyp. Polyp grew out those buds, and the little medusas come out from those buds, like this. Okay. Then medusa get mature and spawn and die. Okay. So you can see this is sexual phase and this is asexual phase. And they are between about 85 to 98% water. Even though most of their bodies are water, they still have complicated structure like this. Please pay attention to those with underlines. We're gonna use them later. One is its umbrella. It's like an upper side of its bell. This area we call it bell because of its shape. Then radial canal and circular canal, which is also known as ring canal. And these two canals is related to digestion. How about polyp? You can see I only point out two structures of it. One is endoderm. It also related to digestion. And the other is parasite. Parasite is like armor for polyp. It protects his body. Okay. Here comes our main character today, Turritopsis nutricula. I'm gonna call it D in the rest of my talk. And it looks like a lighthouse. I don't know. And they're about 5 millimeters. It's like a nail, that big. And they have special life cycle. Okay. Here is a normal medusa's life cycle. And the T can directly, when they get old, they can directly turn into polyp to avoid natural death. And because it's a condition which from old to young, so we call it reversal. And this is what we want to stop. Okay, for study list, we have 3 main questions. One, in which case will it reverse? Or how to make them reverse before they get old. You know they reverse when they get old. So before they get old. 2, what's the specific process of reversal? 3, which part is used to control the reversal? Okay, for each question we design an experiment for it. In which case we do environmental stress test, such as higher temperature or something, and the specific process we just observe with the microscope. Then which part we cut and rear the tissues independently. Okay, like this. Okay, material and method. About 4,000 T were caught in the Gulf of Naples. And the time is about June to October in 1993 and 1994 here. Okay. And they will be reared in 40 beakers. This is beakers, which is about 500 milliliters. And each beaker has 100 individuals. The temperature is about 32 degrees and they are feeding with tiny shapes. Okay. Okay. The experiment 1. Environmental stress test. We have 4 groups. 1 is starving, 2 temperature, 3 salinity, and 4 control group. Each group we have 10 newborn medusa. And for starving, we just give no food, and the temperature will swiftly change to X degree like this. You can see directly 1 degree, directly 2 degrees. No matter. Okay, for salinity, we just add pure water. And all these tests are be done when they are young, because we know they reverse when they get old. Okay. And for observing, we use a fluorescence microscope. You can get a picture like this. Okay? Then isolate each part. This is the part we cut. Okay, so you can see A to I, 9 parts. Don't worry, I listen down. And you can see the structure we talked before. Remember? Okay. Okay, and for all these parts, there will be 2 results. One is successfully reversed, and 2 is dissociation, which means that. Okay, here comes our results. Stopping, it reversed, and the temperature is reversed too, and it's about 5 degrees upper or lower. And the salinity is still reversed. It's about 10% water. The salinity is 33, and the control group, it did not reverse, but it's still reversed when it gets mature. Okay, then observing what we got. This is what we see. You can see from a Medusa, it get regress and become polyp. Here is a clear version. You can see Medusa, regression, settlement, transdifferentiation, and the polyp here. Okay. How about our experiment 3? We got 4 successfully reversed part. Is D, G, H, and I. You can see most every part have its umbrella and the canal. And no matter it's radial canal or ring canal, canals at the same time. So we can get the formula. Its umbrella plus any canals equals success. Okay, discussion. So why does it reverse when facing stress? It's like a mechanism for that. Like we touch the hot part, we're gonna get ouch. It's like that. It's a mechanism for them when they're facing death. And it also can copy itself through asexual regeneration to maximize the probability for living. Okay. 2. Why is that it only success when including loss structure and its umbrella and canals? It's because its umbrella will become the parasite of the polyp, and the circular canal or radial canal will become the endoderm of polyp. Here. Oh wow. There's the. That's okay. And number 3, will it die? No, but just no natural death. That means it will not die because of its age, but it still die in main condition like being eaten or disease. Okay, conclusion. Also, of Turritopsis nutricula is confirmed to be a part of their life cycle. And for its reversal, its umbrella and the circular canal and the radial canal are needed. Thank you.
8 minutes 54. Scott. Jesse. Sally. Sally here. Amy. Daniel. Okay, starting with Scott.
Why you choose those 4 parts or 3 parts? I forgot. So to cut. So why you don't cut other parts?
Because mean here? Okay. It's because it chooses the different cell type and the essential structure of the Medusa. Like this area is about digestion and circular, and like tentacle here, just important structure for it. So I don't know it's. I don't know.
Next.
Okay.
Jesse.
About environment stress test. Why do you choose these three groups? Why is temperature or salinity or about their foods? Why is this?
Okay. They didn't mention this, but I think it's because it's the most important index of in the sea. So if you change this lake may perhaps have some change. Yes.
Next is Emmy.
You said that they don't die in nature, but their cells and their DNA will get old. So why don't they die?
I'm sorry.
Repeat again.
You said they don't die in nature, but their cells and their DNA will get old. Right? So how can they never die in nature?
Never die in nature. Actually, it just don't die, and DNA is totally the same. It's the same, but it regression and become polyp again, and buds, and come out Medusa. Then become polyp again, like this. Totally the same DNA.
Last one, Daniel.
Can you show me the reverse picture
Reverse picture.
from Medusa to not that.
Result.
Yeah. So it's paper they do examine those Medusa and polyp, their DNA are the same.
No, but they did not do something sexual based. So it's the same. It's like a sexual. Yes.
So is that your question? Volunteer question, Emmy. I can only take 3. Okay. Skip those who has asked. Okay. Yang and. Okay.
Why do you choose to reduce salinity test, or what if you increase the salinity?
They didn't mention, but I have searched. I think another paper said it will reverse till around 9 PSU upper. So maybe it can fit high salinity bit better. Yes.
Yeah.
say if the temperature become too high, they will reverse.
Yes.
So, but after they reverse, the temperature is still high for them.
Yes. Yes.
So why they reverse at first?
It's like I gonna die today, and you say no, not today, and reverse.
To experience newborn individuals, unless sexual from sexual excitation.
No, actually, all the little Medusa are all the little Medusa are from a sexual phase. It came from those spots.
Okay.
Yes.
Thank you.
Okay. I think you will also agree with me. It's a very entertaining talk, and yeah, yesterday when I was reading the paper, I was thinking, wow, this is a really good story. I think it shows. So come back to his presentation. I think he also presented the story very nicely. So he talked very smoothly, and his slides were easy to follow with short sentences and many pictures. And actually, he also, I think, I'm not sure. Did you draw this yourself? No, it's from the internet. Okay. It's, I was thinking because the original paper was like graph was like this. So he simplified this, but at the same time, he also missed some interesting details. So because this time you didn't provide enough, or you didn't make it 10 minutes, I would suggest that next time you can probably talk more about these details, because I think when I was reading the paper yesterday, it was really interesting, like this. Like the jellyfish could even turn itself inside out. It's a little bit like that. So these are very interesting details, and I think next time please provide more details, because these are not complicated and these are very interesting animal behaviors. And also next time, if you still couldn't make it 10 minutes, you can probably add some implications, because since the discovery of this animal, I think it has been the target of many biotechnology studies. I think it's super interesting. And come back to his talk. He presented nicely with, in his talk there was always a sense of humorous. I think you feel that. I think I really acknowledge that your talk is humorous, because I think it's a very special properties. And he also used a lot of body language and nice artwork. And he also used some very nice analogy lately to talk about the concepts in this paper. Like he talked about like some hotspot or something. Okay, so overall I think it's a really nice talk. The only weakness is it's not 10 minutes. So next time I'm sure you can do it. Okay, let's thank Annie.